Good afternoon, distinguished guests, ladies and gentlemen. Selamat sore and herzlich willkommen. Allow me to introduce myself. My name is Dr. Stephanie Jirgans. I'm the lead researcher from the ASDS, but most of the time people just know me as Dr. Nani.
Yeah, hello, good afternoon, everyone. I'm Novitria Haryani. Together with Dr. Nani, I honors to become your MC in this afternoon. And today is not a more only symposium, it also reflect a shared visions to make a genomic more affordable, inclusive, and also impactful for Indonesia. On behalf of the organizer, our great pleasure to welcome for all of you here in the Jakarta today in BioAI Genomics Symposium.
We are gathered at the meaningful moment advances in next-generation sequencing are lowering costs, expanding discoveries, and opening new possibilities for healthcare systems. For Indonesia, with its huge scale, diversity, and growing ambition, this creates a real opportunity to build a genomics ecosystem that is not only advanced, but also collaborative and accessible.
That's why today is matter. The symposium brings together innovators, clinicians, scientists, policymakers, and partners to explore how genomics can be strength in public health and also expand the access in the precious medicines. And today is not just about the presentations, it's about the sparkling news idea and the building the meaningful collaborations. And if the experience can any guide, and it most valuable for the discussions and may happen during a coffee break. We hope today Ignite is not only discussions, it's also a lasting actions.
Before we begin, we would like to thank all organizations, organizing and supporting partners, which is YSDS, Ultima Genomics, the U.S. Embassy and U.S. Special Services, Commercial Services, apologies, Twist Bioscience, the Ministry of Health of Republic of Indonesia, and the Ministry of Higher Education, Science and Technology of the Republic of Indonesia.
We today spark not only for the discussions, we also meaningful collaborations. As gentle reminder, please kindly place the mobile phone into the silent mode. Thank you.
Dear Excellencies, distinguished officials, honored speakers, and valuable partners, ladies and gentlemen. We truly are grateful for your presence today. It reflects a shared understanding of advancing genomics and precision medicine requires collaborative across sectors. We are honored to welcome
Mrs. Indri Rosmialti MSc APT as Head of Balai Besar Biomedis and Genomika Kesehatan, Ministry of Health of Republik Indonesia, Professor Dr. Matt Trianggono Ahmad as Technical Advisors, Ministry of Higher Education, Science and Technology, Mr. Eric Xu as Counselor of Commercial Affairs in U.S. Embassy Jakarta, Dr. Vincentius Simon W. Budianto as Chairman of Yayasan Satria Budi Dharma Foundations, and also the President Director of Panin Bank is Bapak Herwidiatmo.
Dr. Stephen Lee, the Sales Director from Ultima Genomics, Professor Jong-il Kim, MD, PhD, Seoul National University College of Medicine, Dr. Peter Chai, MD, PhD, and CEO of PDC4 PM Taiwan, and Dr. Jim Zhang, Field Application Scientist from Twist Bioscience.
And Johannes Robini Marianto, S.Phil M.A., Brigadier Purnawirawan Dr. Randa Sumirat Widiatmo, MSc, a superior of the House Assayan Dominic Pontianak, and all invited guests, partner, colleagues, and a friend. Thank you for joining us. And your presence reflects the growing importance of the collaborations to building of the stronger genomic ecosystem in Indonesia. And it's now to honors to invite the first opening speakers, representing of the Ministry of Health in Republic Indonesia, Mrs. Indri Rosmialti, MSc APT, a serve of Head of the Balai Besar Biomedis and Geomedika Kesehatan. Please join me as warmly welcoming Mrs. Indri Rosmialti to deliver her opening remarks.
Okay, so thank you again for the invitation. So, distinguished representative from the United States Embassy, and then also for the chairman of the YSDS, and the representative from the Ultima Genomics. And I hear from the MC there are lots of speakers, lots of invitations. The professors, researchers, lead from academia, industry, students, ladies and gentlemen, a very good afternoon to you all. So it's my honor to convey a warm greeting from the Minister of Health. He cannot be here today because he has another agenda. And then I think he would like to give an appreciation to this event, because this event is a good event to promote the research and also the collaborations between the academia researchers as well as industries and also the governments. So this forum reflects a shared global commitment to advancing health system through genomics, and also I think AI is now invading the world, so we need to cope with that. So distinguished guests, so globally we are witnessing a paradigm shift in healthcare, so from curative into the predictive, preventive, and precision-based approaches. I think this is where the genomics comes. So we can start from the screening and also from the preventions. And the Red-Scale Genomic Initiative have demonstrated the real-world impact of integrating genomics into the health systems. So for examples, we have the UK Biobank, now with over the 500,000 participants, has generated one of the most comprehensive biomedical data sets. So it's not only the data from the sequencing, but also the clinical data. These resources has enabled thousands of scientific publications and significantly advanced our understanding of complex diseases while supporting the development of the PRRs, for examples, for early risk predictions and preventions. And also, I think we can see from our neighborhood countries, Singapore's, with their initiative called PRECISE, it's offer precision medicine cohort, which is focused on the general populations, have demonstrated how integrated genomics, clinical, and lifestyle data can improve disease stratifications, enhance early detections, and support more efficient healthcare delivery. And also one more country in our regions is Thailand. Now they already reached 50,000, and I think they just published in one of the paper recently about their 500,000 achievement. So this program has contributed to improving diagnostic accuracy, and also I think they're focused also in rare diseases and pharmacogenomics implementations, ultimately enhancing clinical decision-making and optimizing treatment outcomes within the healthcare system. And I can tell you that Indonesia is fully committed to these transformations. So testing in 2022, I think, in the Ministry of Health, we are advancing a comprehensive strategy to leverage the biotech, the AI, and also advance the medical technology as the key drivers for the health system strengthening. So this initiative called the BGSI, the Biomedical and Genome Science Initiative, now we're already 2.5 years old. And this program is in Ministry of Health priority programs as in embedded with our national strategic plan called the Renstra, Kementerian Kesehatan, with a clear performance indicator. So we have the indicator for the next 5 years to ensure the measurable impact and sustainable implementations. So these innovations are enabling a more data-driven, personalized, and patient-centered care in line with global scientific evidence. So the precision medicine has been shown in improve in clinical outcomes by tailoring interventions to individual genetic profiles. I think one of the limitations of our regions is about how the data of our regions is represented in the global community, because like based on several publications show that our population is only, I think, less than 1%. I think from the last workshop that we had in the Ministry of Health, it shows that we are not included in the Southeast Asia even, so we are included in others, which is less than 1%. So we need to ask the questions. So are the database that is currently we have at the moment, which is based on Caucasian population, is it really reflect our data is Indonesian, because we are 280 million people. So that's why now the Minister of Health, we are trying to make this not as a one-time project, but as the sustainable program. So currently, we have quite a meaningful progress in the 2.5 years. We have around 22,000 participants recruited from diseases. And also from the general populations. For example, we have from the TB patients, we have from the stroke, cancer with the 4 cancers, we have from also diabetes, we have also from the mental health, and also it's 9 diseases that cause a high burden in Indonesia. And now we sequence around 17,000. And of course, I think the main key is how we delivered the result to the patients. So because now the regulation is not there yet. So now we are carefully trying to have a discussion. Hopefully in the end of this year, we can have really good regulations how to give the result back to the patients. Example for the pharmacogenomics, or the result from the predictions of the cancer, for example. Because like for the pharmacogenomics, we need, because this is the key is from the clinicians, right? Because they are the one that can the task, and also how the roles of the clinicians, how the roles of the clinical geneticists, how the role of the genetic counselor in this ecosystem, because I think they are really important as well. But we haven't put this into regulation. Who will be the one responsible to give the result back to the patients? I think that can be also discussions for us. And we also now have 9,000 PGX reports generated. And I think in the end of the day, what's the point of collecting the data? Of course, we need our data to be utilized by the ecosystem, right? So currently, we are now developing our trusted research environments. So hopefully, in 2028, hopefully in the beginning, so the ecosystem can use the data through the trusted research environment. This is already implemented by the UK Biobank. So what we're doing is not, we're doing based on what we wanted, based on what the Ministry of Health wanted. But we had several benchmarking, and also we're currently asking for the expert from those specific countries to ask us to help us what actually they did. What the advantages of being not last countries, but I think the last countries that are starting this genomic initiative is that we can learn from them. So we don't need to repeat their mistakes. So I think that is one of our advantages at the moment. And also now we are currently developing the first gene bank in Indonesia. So hopefully we can have it in, I think, end of 2026, and it can be rolled in the beginning of 2027. And of course, for the accessibility, equity, fairness, we already think about this since the beginning, how the ethic, legal, and science implications, we already talk about this in the beginning as the white papers. And I think the main key is that how this continues improve the quality, effectiveness, and equity for healthcare services for Indonesian peoples. So we believe that no country can achieve this transformation alone. And the success of the initiative that I mentioned previously is about, we need to highlight about the importance of strong, sustained, and cross-sector partnership. So we therefore highly value collaborations such as those fostered in today's symposium, including with the partners like now, Genomics, YSDS, and all these people here, and also all of you guys that's already attending this event. And then Indonesia remains open. We welcome new innovations that can accelerate the health transformations, and also committed to building an ecosystem that prioritizes inclusivity. So there is no silo again. So we need to work together hand in hand through strengthening partnership across sector and borders. So through collaboration, innovation, and shared responsibility, we are confident that we can build a more resilient, equitable, and future-ready healthcare system. Thank you very much, and good luck with the symposium.
Thank you, Ms. Indri. And what an amazing number, already 17,000 sequenced and 30,000 by the end of the year, right? Hopefully, yes. Okay, our next opening remarks will be delivered online by Professor Dr. Tri Hanggono Ahmad from the Ministry of Higher Education, Science and Technology of the Republic of Indonesia. The floor is yours.
Well, okay. Thank you very much. Hello?
Yeah, we could hear you earlier.
Okay.
Maybe you just start. We
Okay,
don't
thank
have a
you.
picture yet, but just go ahead. They're trying it out.
Okay. Okay.
Yes.
Ladies and gentlemen, distinguished, good afternoon. First of all, I would like to share our greeting from the minister because of the agenda. He cannot join this meeting directly. Apologize for that. Of course, we would like to appreciate the organizing committee for organizing this kind of meeting. For higher education, it's a very meaningful meetings. As the said, that we have a big challenge in developing our genomic study. Many universities has capacity for this, but again, we have to work hand in hand in collaboration because first, it is not easy technology for this. Not all university has kind of this high technology, but by working hand in hand in collaboration, I think we can achieve what we would like to have in the future. Indonesia is very rich for their biodiversity, especially for this genomic future. So again, we would like to appreciate this. Collaboration between university, industry, and also the government is very important for this. So we would like again also to appreciate the participants for many universities who attend this meeting. And we hope that industries can also support the university because they have a lot of expert in this aspect by collaborating something like this, not only for Indonesia, but I think with the President, again, congratulations for the organizing committee for organizing this meeting. We hope that we can have a very fruitful seminar today. And after this, we hope that the further collaboration can be achieved by each party who participates this time. Thank you very much again. Have a fruitful seminar. Thank you.
Okay, thank you, Professor Tri, for inspiring remarks. And our next speaker represents the key partner in strengthening the international collaborations. Mr. Eric Xu is the counselor of commercial affairs at the U.S. Embassy in Jakarta, within over 20 years of experience across of Asia in diplomacy, trade, and finance. He currently leads of the U.S. Department of Commerce in the program in Indonesia, advancing partnership and also economic cooperations. And we have heard today expanding genomics is not only the technology, it's also for the global collaborations. Ladies and gentlemen, please welcome Mr. Eric Xu.
Good afternoon, everyone. It's such a pleasure to welcome you all to this afternoon's BioAI and Genomics Symposium, hosted in partnership with our partner, Ultima Genomics. Please allow me to take a moment to extend our appreciation to the following distinguished guests and speakers. First, Ibu Indre from the Ministry of Health And you all heard from just a moment ago, Professor Tri Hangano from the Ministry of Research, Technology and Higher Education. Also, many thanks to Ibu, Ardina, and Dr. Vincent from the YSDS Foundation, and Jimmy Zhang from Twist Bioscience. Also, of course, the many distinguished scientists, professors, and researchers in the room today. Thank you for your presence. Again, my name is Eric Xu. I'm the Commercial Counselor for Commercial Affairs at the U.S. Embassy here in Jakarta. Now, at first glance, the symposium this afternoon appears to be just one of the many gatherings on genomics. But the scale and the ambition of what you'll be hearing and what will be the expert will be discussing today are fundamentally different. We are entering a new era where data is no longer the limiting factor. Rather, it is how we harness that data and deliver meaningful health outcomes. Indonesia stands at a unique inflection point. Its large, diverse population and the government's focus on healthcare transformation create new potential for population scale genomics. At the same time, challenges remain in access, infrastructure, and affordability. The United States government is actively supporting the development of genomics and precision health through agencies such as the U.S. National Institutes of Health, NIH, and the Centers for Disease Control, CDC. The U.S. government supports research, standards, and global health collaboration. We also support partnerships that accelerate innovation and responsible data use. Public-private collaboration is central to scaling genomics technologies and expanding access. Ultima Genomics is an example of U.S. innovation contributing globally. Its technology helps lower sequencing costs and enables genomics at population scale. These advances support better research, earlier diagnosis, and more precise use and care. Ultima Genomics also lays the foundation for the emerging BioAI era. Today, we will witness the signing of an MOU between YSDS Foundation and Ultima Genomics to advance genomic sequencing across Southeast Asia using the UG200 platform. The partnership aims to sequencing up to 1 million genomes, expanding access to affordable, large-scale clinical genomes across the region. This is more than just a signing. It's a commitment to action, a commitment to ensure the benefits of precision medicine reach the broader population. The United States stands ready to deepen collaboration with Indonesia. Together, we can advance innovation, strengthen health systems, and improve outcomes. On behalf of the U.S. Embassy here in Jakarta, again, thank you for joining us today. We look forward to a productive exchange and to deepening our cooperation in support the advancement of genomic research and precision health. Please do not hesitate to reach out our team here, the commercial team at the U.S. Embassy in Jakarta, should you require any assistance. And I wish you all a very productive and impactful event this afternoon. Thank you.
Thank you, Mr. Xu. Dear ladies and gentlemen, now we come to a very special moment of today's afternoon. If the staff could please now bring the table to the middle as well as move the speaker board.
Yes.
Maybe we just give them a quick moment to get everything ready. Because this afternoon, the MOU signing remarks an exciting step forward for genomic collaborations in Indonesia and across Southeast Asia. It reflects a shared vision to make high-quality genomics accessible, scalable, and affordable. Building the partnership, the infrastructure needs long-term impact.
With that, I'm pleased to invite from WSDAS, Ibu Erlina VF Ratu, and the chairwoman, and also Dr. Vincentio Simeon Widianto, and from the Ultima Genomics, Dr. Jason Kang, to the stage, and for the official signing the ceremony between WSDAS Foundations and also Ultima Genomics.
So today's signing reflects not just collaboration, but a commitment to act and impact across the region. Therefore, if I can ask the parties, please sign right above your name.
Yeah.
And now please exchange the folders.
So, now it's time to take a pictures before we leave from this stage. Yeah, in front of this table.
Adi, thank you. And again, congratulations to both institutions on this important milestone. And we're looking forward to setting the impact of this partnership across Southeast Asia.
Yeah,
Thank you.
Yes,
The
please.
staff could now
Just
please-
remove the table.
So maybe we just wait a moment and then we get into the next part of the session.
Take a picture with Bu Indri because Bu Indri wants to this room. And we need to Bu Indri, we invite Bu Indri to the stage. Pasti itu ya. Boleh, sekarang tayo. Now I want to invite Ibu Indri Rosmiati to the stage to get us some souvenir from us and take a pictures to our. Please Ibu, Jason or Ibu. Ya, yes Bu, silahkan. Yeah, from the doctors, Dr. Vincent and also from Jason.
First time.
Yeah. Take a look at camera first, Bu. Yeah.
Thank you.
Thank you, Ibu. Thank you for the opportunity to come to this event. Okay, and now we need to continue the affordable genomics for the accessible and now we bring to scale the system for the collaborations. And the first, our presentations to highlight the technology of the sequencing, economic, and also from the Ultima Genomic, how to drive a lower cost for whole genome sequencing in the population scale. And also let's keep our questions until the end. So we will have a Q&A in the end of the sessions and will be collective at the end.
So the UG200 system delivers high through output and cost effectively, supporting large-scale population genomics, pharmacogenomics, oncology appliance, and BioAI data generation. So therefore, please welcome Dr. Stephen Lee to the stage, the Science Director of Ultima Genomics. Stephen has earned his PhD in genomic science from the University of Washington and leads the genomic strategy adoption across the APAC region. Please welcome Dr. Lee to the stage.
All right. Hello, everyone. I'm Stephen Lee. I'm Sales Director for Genomic Applications in Asia for Ultima Genomics. Thank you so much for joining us today. Today, I'm going to be sharing about how Ultima Genomics is powering the BioAI era in genomics. But before I delve into this, I want to first make sure that we're on the same page regarding why AI is necessary and essential in biology. As you know, there are 3 billion bases in each copy of the genome, and we have two, one from each parent. And for each base, you have 4 different possible nucleotides, A, C, G, and T. So if you think about it, if our sequence was truly random, you will have combinations that will amount to 4 to the power of 6 billion. That's actually more than the number of atoms in the entire universe. Thankfully, our sequence is not random. It's more like the language. And as you know, AI models like ChatGPT and Gemini, these are all called large language models. So AI model is super good at interpreting this type of language. And that's why it's natural fit for AI to be used in biology. But it's not just DNA sequence, right? We're starting with as a single cell, but we differentiate, replicate, and grow into 37 trillion cells in our body. And depending on the organ or tissue type, you have thousands of differentiated cell types, epigenetics, gene expression, etc. And by the time we hit our age at 40s or 50s, it's been estimated they will be accumulating about 10 to the 17th mutations. That's a lot of mutations. And that's why, we are essentially walking mistakes. Worsening the situation, if you look at the basic unit in biology, which are cells, they're incredibly complex, and there's no mathematical equation that still can reliably predict the influx and outflux of a cell. That's why at Ultima, we truly believe that AI will do to biology what math did to the physics. For example, Isaac Newton, when he discovered gravity by observing an apple falling up a tree, physics used to be observation-based science. But as our mathematics got better and better, physics became more of a precision physics, and we can make a lot of accurate predictions. That's why we have computers, smartphones, the screen that you're looking at, airplane, etc. Similarly, biology, researchers, they used to look at cells under the microscope, making observation again. But with AI, we believe that biology will become more precision and prediction-based. The only problem here is that AI requires a lot of training data, and ChatGPT and Gemini, all these foundation models were trained on publicly available free internet data. Biology, somebody has to pay for it, and somebody has to generate all that data, because right now the existing data sets are not enough to train the data or the AI models reliably. So when I'm talking about data, training data, I'm talking about sequencing data because as many of you all know that sequencing can be used for identifying genetic variation for your genome, but sequencing can also be used for measuring RNA expression, epigenetics, proteomics, circulating tumor DNA, whatever. All of these can be measured by sequencing. So now sequencing is more like a computation tool like CPU and GPU in compute. So at the end of the day, it's a sequencing cost that really is the core of whether or not we'll be able to achieve the ChatGPT moment in biology. Thankfully, sequencing cost has been decreasing, but at Ultima, we believe it's not fast enough. That's why our CEO, he's not even a biologist. He used to work in semiconductor industry, applied materials, and he transitioned to biology because he wanted to lower the cost of sequencing really fast so that we can enjoy AI moments earlier. So the graph that you're looking at right now is our competitors' sequencing cost. Right at this moment, their latest and biggest sequencing cost, I'm not going to name the company and the platform, but it's roughly $200 per genome, 30x whole genome sequencing at 100 gigabytes of data. Ultima, when we first launched in year 2024, we came out with $100 genome. So we were already 2 years ago, half of what our competitor was able to provide. But just in 1 year, so that'll be last year, we were able to reduce the cost even further down to $80 genome. And this year, it was mentioned earlier, the Ultima release launched UG200, our new platform. And we haven't announced exactly how much it's going to be, but you can see the chart, kind of guess how much it's going to be, around 60 to 60, definitely below 70, because it's not going to go up. And we're not even stopping here. If you look at our roadmap, we are continuously lower the cost down to $10 genome. At that point, sequencing will be as ubiquitous as PCR, and sequencing costs will no longer be the bottleneck for genomic innovations. That's the kind of world that Ultima is dreaming and trying to realize that dream. So the $1 million question here is how is Ultima doing that, right? Reducing the cost of sequencing, because a lot of people ask, aren't you just lowering the cost, bleeding your cash, and maybe your company going to go bankrupt in a few years? That is definitely not the case because we are using wafers as a substrate for our DNA sequencing. And for those who are familiar with next generation sequencing will appreciate that other competitors, they are using what's known as flow cells. And flow cell is a great technology. It's a great piece of, a sophisticated machinery, but at the same time, it's really expensive to manufacture this precision manufacturing flow cells. That's why Ultima, we use wafers, and wafers look like this. I brought a wafer myself. This is the exact wafer that we use for sequencing DNA. But it's also the exact same wafer that companies like Samsung Electronics or TSMC, all these fabrication companies are using to create computer chips. So the reason why we are using wafers instead of flow cell is because wafers, even though so for those who are not familiar with wafers, wafers are the substrates for computer chips, semiconductor industry. And the entire industry of semiconductor has been perfecting this technology for decades. That's why even though this is super sophisticated technology, each wafer is actually incredibly cheap. You can right now Google how much each wafer costs, but it's really just price of a cup of coffee. That's why we're able to lower the cost so dramatically, yet stay profitable. So instead of manufacturing our own wafers like our competitors, we just purchase wafers off the shelf. So that's how we're able to maintain this very cost-effective sequencing reagent and mechanism. Not only that, we are also borrowing many cool tricks from semiconductor industry, like spin coating. So we actually spin our wafer, and that centrifugal force delivers the reagent to the entire surface of the wafer very uniformly and very efficiently. So our sequencing is actually really fast because the wafer spins like an LP record. Also, now that Ultima is kind of removing the cost of sequencing from the equation, that it's no longer the bottleneck in genomic innovation, we identify new bottlenecks popping up, one of which is computation. Because we are generating data at a scale that's unprecedented, it's really important that we have architecture that can support that type of data generation and scale. That's why we're working with companies like NVIDIA to incorporate GPU-based accelerated computation. Another bottleneck that's popping up as the sequencing cost goes down is automation and scale. Because as sequencing cost goes down, researchers, they want to increase their sample size. That means you have to run a lot of samples, high volume. And if there's no automation to back up those high volumes of sample, then your operation is not going to be feasible. That's why when we built our DNA sequencer, we designed it such a way that it's highly automation friendly. What I mean by that is our sequencer, first of all, you can run two of these simultaneously. And then on top of the two wafers that are already running, you can also store up to 10 wafers and 10 sample pools in the sequencer. And it's got a robot arm inside. So after a wafer is done, it's going to grab next wafer and start the next run automatically for you, so that the operator doesn't have to be standing in front of the machine to manually start a new run. That's one big distinction between Ultima and other competitors, where for other machines, operator literally has to be there, start a run, and after run is finished, he or she has to do wash manually. But in our case, it's like a DNA center, just like there are data centers, all automatic, and operator can start a run on Friday, and the machine's gonna run automatically for you on Friday, Saturday, and Sunday, and the researcher or the operator can come back on Monday to pick up where things were left off. So it's truly 24/7 operation. So this is our analysis deck. So you see we're starting from sequencing to primary, secondary analysis. In this case, primary would be base calling and secondary would be like alignment. And then tertiary analysis would be variant calling. And every step of the way, we are using GPU-based acceleration in order to handle the amount of data that we are generating. And I want to emphasize that even for tertiary analysis, like variant calling, we are working with NVIDIA and Google, because Google developed deep variant. Google developed many genomics innovations like AlphaFold, AlphaGenome, deep somatic, deep variant. I mean, they're going crazy. But basically, it's really good fit because they are also really good at analyzing big data scale informatics. So I want to briefly touch upon the progression that we have made so far. Again, we are a relatively young company, only going commercial year 2024. But if you look at the numbers here, 2024, each wafer was able to generate about 6 billion reads. And that was a $100 genome. And just 1 year later, we came out with a new chemistry, UG100 Solaris, and each wafer was able to generate 10 billion reads, and it was $80 genome. And this year, again, we haven't announced the exact pricing, but each wafer can generate now 20 billion reads. So you can kind of get the idea of how Ultima is able to reduce the cost of sequencing by increasing the throughput per each wafer or per each experiment. Also, I want to give you a sense of the capacity of our sequencer with all that automation and data generation, right? So in terms of 30x whole genome sequencing, 1 unit of our UG200 sequencer can generate 60,000 genomes per year. Compared to other competitors, our major competitor, their platform can generate 20,000 genomes per year. So even 1 machine is equivalent to 3 machines of our competitor. So I just wanted to let you know. So I want to switch gear a little bit and talk about science. So what are we going to do with all the data that Ultima is going to generate, right? So I think, and this was briefly touched upon earlier, speeches, but basically with AI-enabled biology, we have 3 categories: bottom up, top down, and dynamic. What I mean by bottom up is that we're starting with measuring proteins and single cells, organs, and then go up to a human. And top down is, completely opposite. You start with population level, UK Biobank, Precise for Singapore, and then you're trying to link your genomic information with phenotypes or EMR data. And finally, dynamic, meaning you're only looking at 1 person, but longitudinally. So repeated testing, whether it's a cancer monitoring or infectious disease, whatever. So I want to first start with bottom up. So we've been very active in single cell projects. So this is one from BioHub, which is part of Chan Zuckerberg initiative. So Zuckerberg is the founder of Facebook and Meta, and they have been giving out a lot of fundings for single cell projects. The latest one is billion cell project, where they're literally trying to sequence gene expression profile of billions of cells. And you may be wondering at this point, then what's the good with sequencing billion cells? They're trying to build a foundation model for predicting gene expression response for all kinds of conditions. So their AI model is not specific to a specific disease or tissue or organism even. It's completely generalizable, physiology-aware model. That means pharmaceutical companies, for example, if they develop a new drug, traditionally, they will do a lot of wet lab validation experiments, like mouse model, for example. But using this foundation model, they can actually predict in silico how their drug's going to affect downstream gene expression profile and also pathways that are involved. This is more recent collaboration that was announced a couple of weeks ago, the Trillion Gene Atlas. This is a collaboration between Basecamp Research, Ultima, PacBio, and NVIDIA, and Entrophic, the developer of Cloud Code. Basically, the idea is that Basecamp wants to capture all kinds of genetic variation that's already existing in the environment. So what they do is that they actually go out in these extreme sites like volcano or iceberg and taking environmental samples. You may wonder why they're doing that. Basically, they're trying to capture the spectrum of DNA variation that already exists in the environment because, for PCR, for example, you're using Taq polymerase. And do you know where the polymerase was found? Hot springs, because the polymerase needs to endure that hot temperature. So that type of extreme locations actually give, like a microbiome, microbes and viruses, phages to come up with really creative way of overcoming these natural limitations. And those sequences actually are used to find therapeutics. So Basecamp has already validated their foundation model by identifying therapeutics, followed by wet lab validation. And what they're trying to do now with Ultima is that they want to expand their training data to come up with more creative therapeutics. Really exciting project. So I want to switch gear. Now, top-down approach. So, you heard about UK Biobank and Precise. I mean, all these projects, because the sequencing cost is the problem, they tend to use like SNP array or targeted sequencing, like whole exome sequencing. But if sequencing cost is no longer the problem, they can do whole genome sequencing for all of the samples. And at this point, I want to emphasize it because I know some of you might have questions about the quality of Ultima data because we are relatively new. So the data that I'm showing here is genome in a bottle benchmark results. And for those who are not familiar with GIAB, basically every time there's a new sequencing manufacturer or new platform, well, what companies usually do is sequence reference material DNA, Coriell samples, and then perform our own variant calling, and then compare our variant calls against true sets. So this is a F1 score table against the true sets that have been published. So this is really objective way of assessing accuracy of a platform. And, basically all companies are doing that. And you can see our accuracy is really good on the paper, but also in real life as well. And with these numbers, and you can actually download all these data sets because these are publicly available, and you can try to replicate these results as yourself as well. And I'll be happy to guide you on how to do that. But with these data sets, you can do head-to-head comparison with other competitors, as you can see here. And you can see our numbers are on par with our competitors. So you can be sure, basically, why pay more for the same quality of data? So what are you going to do with all the, whole genome sequencing data that is not SNP array and that whole exome sequencing? And again, pharmacogenetics was mentioned multiple times, so I'm not going to spend too much time. But no matter which ethnicity or population you're looking at, you will see a chart that looks like on the left side. Usually we expect distribution to be a bell curve normal distribution, but it's not the case for pharmacogenomics. You will always see this type of multimodal distribution where you have poor metabolizers, ultra rapid metabolizers. This is actually for CYP2D6 enzyme, one of the most important pharmacogenes that's known to metabolize about 25% of the prescription drugs that you will find in a drugstore. Really important gene. And you may wonder then why there's so much variation in the first place. And that's because the gene is located in a region that is really complex. As you can see, the gene is right next to a pseudogene, CYP2D7. And long story short, they have all kinds of structural variations, like gene deletions, duplications, hybrids. And then you also have to worry about SMBs and indels. So with whole exome sequencing data or SNP array, it's just forget about it. You're not going to be able to reliably characterize these variations using those technologies. But with whole genome sequencing, you can actually comprehensively target these variations and come up with a report that be really are comprehensive and accurate at the same time. That's why a lot of big projects, like all of us in the United States, Precise from Singapore and UK Biobank from UK, they're all starting with pharmacogenetics because this is kind of the low-hanging fruit in precision medicine. Switching gear, I also want to talk about a very specific example where one user was doing SNP array genotyping but converted to whole genome sequencing using Ultima. So MyHeritage is the second biggest direct-to-consumer genetic testing company in the world, next to 23andMe, and they are processing 1 million people per year. That's one for each month. That's 100,000 samples per month. That's a lot of samples. And they were using SNP arrays up until last year, and they switched to Ultima sequencing. It's whole genome sequencing, low pass, but still whole genome. So their resolution of ancestry analysis and healthcare reports got boost for whole genome data. Yet the very good news is that end users, the customers, are not paying more. They're actually paying the same price as a SNP array. That really tells you how cost efficient our platform is. I also want to emphasize that UK Biobank has chosen Ultima platform to be the sequencing platform for their largest human proteomics project ever in human history, 600,000 samples for 100 protein markers. So, if sequencing cost is not cheap, then it's really difficult to do this type of multi-omics analysis at the population scale. So that's why, again, we really want to remove the sequencing cost from the equation. So now my last section is dynamics, and more specifically in this case, liquid biopsy. So for those, as you know, our cells are degrading, 2.5 billion cells per year, apoptosis. So cancer cells are continuously shedding DNA to our bloodstream. So that's why, cancer monitoring is really a field that's increasing. Like in the United States, it's being covered by Medicare, etc. So it's becoming a really hot area in genomics. And you can try to detect all kinds of signals from your blood sample, not only DNA, but methylation, proteomics, RNA, fusion genes, etc. And also you can't forget about NIPT prenatal testing as well. But I'm going to focus on cancer monitoring. So cancer monitoring is really, adequately compared to needle in a haystack, because 1 milliliter of your plasma sample only contains about thousands of copies of your DNA. That's actually a really small number of DNA, if you think about it. And a lot of diagnostic companies, what they're doing right now, they're doing panel-based sequencing, right? And these panels, how many genes do they cover? 500 at most. Then you're literally wasting mutation signal outside of those 500 genes. Yes, those 500 genes are really important. They tend to have more driver mutations. But when you're doing cancer monitoring, actually don't have to just look at driver mutations. You can be observing for passenger mutations that are specific to each patient. And those can be actually used to determine the presence of cancer for the cancer patients who just had, treatment, basically. So that's why Ultima, we are kind of replacing panels with whole genome sequencing-based cancer monitoring. The second part is accuracy. Sequencing, we tend to think that sequencing is perfect. It's not. That's why we talk about, Q30 scores. But when you're dealing with cancer monitoring where, mutation is so rare, 1 out of 1 million wild type alleles, sometimes it's really difficult to differentiate sequencing errors versus true biological mutations. That's why your sequencing has to be really accurate. And Ultima actually provides a solution for that. And finally, economics. Cancer monitoring requires repeated testing, and if sequencing is not cheap, then it's not going to be realized. So that's why Ultima, we presented PPM-seq solution. It stands for paired plus minus sequencing, or it's also known as parts per million sequencing. Literally, we are detecting parts per million level mutations. Without going into too much technical details, basically PPM-seq is a variant of duplex sequencing where you are trying to sequence. So you have for a given double-stranded DNA molecule, right? Because our DNA is double-stranded. You're reading both the top strand and the bottom strand and see if a mutation is supported by both strands. Because true biological mutations should have, change in the DNA sequence in both sides. But sequencing errors, DNA damages, or library errors, those are all artifacts, and they tend to occur in one of the two strands, but never both, because statistically it's just not possible. But we're doing this at whole genome sequencing level. That's why actually 50% of our customer base, 50, are diagnostic companies. So we actually started working with diagnostic companies, and we've been expanding into germline whole genome sequencing and other applications. So if you look at, 11 out of 13 leading oncology laboratories in the United States, like LabCorp, Quest, Myriad, all these diagnostic companies have adopted our technology. So I'm going to finish with one example. LabCorp presented this poster presentation last year during AMP, American Molecular Pathology, and basically they have this commercially available whole genome sequencing based MRD solution known as LabCorp Plasma Detect, LPD. And their version 1 was using our competitor, but version 2 switched over to Ultima PPM-Seq because our assay was 13 times more sensitive than their previous generation. I'm not talking about two-fold difference or five-fold difference, 13-fold difference in limit of detection. That's quite significant. And this is just like a start. A lot of other companies are now adopting PPM-Seq, and you'll be seeing more. And I'm really excited to learn more about your specific case of NGS, whether you're doing liquid biopsy or germline whole genome, RNA sequencing. I'll be here to answer any questions at the end, but please find me and talk to me so that we can support your project. Thank you so much for your time, and I really thank your attention. Thank you.
Thank you, Dr. Stephen Lee. I mean, with the price point of like $60 plus, the conversation actually shifts away from an if, but more like really inclusivity. And I think that's the one theme we want to also present in this afternoon or like up to tonight. And that is a theme of, genomic inclusivity.
Yeah, it's my pleasure to invite the next sessions to Dr. Vincent Simeon Woyo Budianto as chairman of Yayasan Satria Budi Dharma Foundations within over 15 years of the experience in biotechnology and genomic innovations. In the behind Indonesia's first oligosynthetizer productions line. And also today he will present about the handling toward genomic inclusivity, roadmap to 1 million sequencing a year in Ignite programs. Please welcome Dr. Vincent.
Yeah. Hello, good afternoon, everyone. I think I will give a little bit presentations about my writings on heading toward genomic inclusivity. But after I present this in English, I will also explain a little bit on our Ignite project. So, I think let's on the other one first. So I'm gonna give a little bit review on no, not that. Yeah. Okay. So this is I make a little bit writing on this. If you want to know the full articles, you can check out on the QR codes. Next slide. Oh, okay, sorry. So as you know, in Indonesia we have 280 million, but in Southeast Asia we have more than 700 million people, and it is like the third most populated region in the world. And the ethnic groups is diverse, but our representations in GWAS is still very low. So on my writings, I divide several countries that already go more further on genomics. Start with Singapore, Thailand, Malaysia, and Indonesia. And there's also the emerging countries on genomics, Vietnam and Philippines. And there's still some early to no capacity right now, like Timor-Leste. So in the writings, I will describe more on what is the initiative happening on each of those countries. In Indonesia, I think I can explain a little bit that we have a dual engine approach. So as MOH initiative that already explained by Bu Indri, we have BGSI or BB Genomica, which is a government lead, which now have more than 70 WGS lab nationwide, and it's supported by UNDP and have some platform. I don't want to compare this, but I just want to show you that we have this program in Indonesia. And other than the government led, we also have Ignite or YSDS project which I'm happy to announce that we are supported by Panin Bank, and Panin Bank is here, the president director of Panin Bank. Can we give a little bit of applause for, yeah. Pak Herwidayanto is the president director of Panin Bank, and I think this is the first time ever in Indonesia a very huge funding from private sector poured in for scientific projects. So right now, I can say that we already have more than 35 sequencing centers with more than 40 research institutes collaborating with us, and there are more than 65 genome research proposal that are running right now. And yeah, we also have multi-platforms, ONT, PacBio, MGI, and Ultima. And actually, if we combine the platforms between BGSI and Ignite, and I think I can say that, 2 days ago, I have a dinner with Minister of Health, and one of his visions is how we can reach 200,000 genome data set in Indonesia. And I tell him if we combine force together, we can easily achieve that within less than 2 years. Yeah. So right now in Ignite we have one T7. We also we'll have incoming Ultima 200. We already have G400s, and we have a lot of Promet ion. So in MOH, they also another T7s which have to 25,000 capacity. And if we combine force, we can reach almost 100,000 per year. So when the UG200 come, we will put it in the government hospital, likely will be Darmais, and I hope we can support the government initiative to achieve 200,000 Indonesian genome within 2 years with UG200. Okay, next slide. Sorry. Okay, so there are gonna be 5 clinical domains that I discussed on the paper. The need for rare and inherited disorder, cancer genomics, pharmacogenomics, reproductive, and this one should be for infectious disease. There's some mistype on it. So you can check on the writings. There's a number that we needed to do sequencing on these 5 domains on the paper. Yeah. So basically we still we have a quite big demand on the sequencing, and on the paper I separated into 3 phases. The phase 1, I think we are having right now. We already have enough platforms to run the phase 1 to help provide clinically procedures, clinical protocol for sequencing. And when I try to compare, I mean, this is also coming from a real experience. When we do in YSDS, we have what we call due diligence. Before we decide which platform that we want to acquire for Indonesia, we will compare it all. And right now, the one that we this is also mistyped a little bit. So with Ultima we can reach 60,000 WGS per year, or with, I think the consumables will be less than this, but we still need to do the extractions, sample collections. So I hope the end cost for everything will not more than 100 for all in WGS service. In the writing I put the recommended fleet strategy to support how we can achieve and serve those 5 domains, clinical domains we want to. And right now, actually, even in Indonesia, we already have at least 3 ultra high throughput we have two T7, one in Ministry of Health and one in our networks. We will bring UG200. I don't know whether, but I hear that Illumina already bring Novasec X to Indonesia. So actually, right now we already have what we need, and the next step is how we can make these tier 1 countries to speak to each other, and how we connect to, because this ultra high throughput will have, to fill those bees, it's gonna take a lot of samples, and I still need to put in more on my writing on the concrete way on how we can talk each other between the hub, so we can utilize the platform maximally. Because right now, actually, the problems with the networks that we have low utilizations, and those low utilizations is because we are more rely on research. We are not like the government-led. The government-led is the population's base. So and the government have, of course, a lot of networks to get samples. But since Ignite project is research-based, so we are based on research. So the intake is quite slow. But within this year, we want to, make it more loose. We will provide more free consumables for universities, so they can use on whatever they want. But if they go within our research topics, they can get even more consumables to do more sequencing. Okay, yeah, this is the scale. I hope in the phase 3, within 7 to 15 years, the consumables consumptions, and it will reach more than 500 million to 1 billion US dollar. And actually, if we see the cost effectiveness at scale, the cost per capita for Southeast Asia is only 0.07 to 0.13 US dollar per capita if we want to make this happen. Okay, so yeah, on my writings, there are 3 essential components. The first is not only short read, but we also need long read, especially for thalassemia, some haplotypes, and complete rearrangements. It's require a long read solutions. And then in Indonesia, especially, it's the workforce, especially for this genomics, still very limited. I mean, for genetic counselor, for the whole Indonesia, I think we only have less than 50 genetic counselor. We need more of it. We also need more bioinformaticians. So yeah, and at the end, since the cost of WGS is getting lower, we are pushing to use this paradigm. We sequence once, query often. So what I see in the future is most likely the array era will be end. So yeah, genomic inclusivity is not a charitable endeavor, but sound, essential health economics. Okay, this is my writings. I want to explain a little bit more about Ignite. So Ignite is our program that's supported by PANIN, and it is a nationwide genomic program for Indonesia. So basically, we are building partnership across Indonesia, most of it with universities. We bring the equipments, we put in the university, we support the consumables, and we give grants for research. So right now, we almost cover all Indonesia. And the one that we still looking for collaborators is on the green one. If you see the green dots, we still looking for collaborators on that area. But on the purple dots, we even already put sequencer there. So right now, as you can see, across Indonesia, we already spread quite a lot of sequencer. Most of it is Promethean from nanopore, but we also bring from MGI, from PacBio, and we also will bring in UG200 from Ultima. Okay, this is all the partner, and I want also to say thank you for our partners that coming here. I see that there's a lot of rectors from universities, and also our startup partners, IGF, also here, and also Willis, the one that assists us on synthesizing. So right now, we also already established oligosynthesizer. In Jogja, we are able to synthesize a short DNA. Every researchers in Indonesia can order primers, and right now I think they already start to do some synthesis on longer DNA. Okay, we also collaborate with the government partner, and I will speak in a little bit in Bahasa because I need to give quite important announcement on Ignite. I'm sorry for those who speak English ya. Jadi Bapak Ibu, sedikit tentang program Ignite, saya ingin memberikan update ke rekan-rekan. Di 2026 ini kita akan siapkan reagen dan flow cell yang akan kita standby-kan secara gratis. Tapi memang jumlahnya terbatas. Kita siapkan itu satu flow cell dan reagen pendukung per bulan. Jadi kurang lebih akan ada 4 flow cell yang kita siapin per institusi. Silahkan itu mau dipakai apa aja. Kita cuma butuh yang akan dimasukkan ke repositori itu cukup abstrak dan data sequencing-nya. Gitu. Jadi kita permudah seperti itu, dan nanti kalau memang rekan-rekan ingin menambah jumlah hibah dari flow cell-nya, itu bisa mengikuti topik yang nanti akan disiapkan oleh tim riset kami. Nah, tim riset kami untuk yang human itu ada Dr. Nani. So I want to introduce Dr. Nani. Dr. Nani is also the MC. She is our lead clinical researchers. Ya, oke. Saya balik lagi ke bahasa Indonesia. Jadi Dr. Nani nanti yang akan urus untuk riset klinis. And also we also have Darwin. Yeah, Darwin is our non-human. I mean, he handled the research on agriculture, medicinal plants, and biodiversity. Yeah. So jadi kayak gitu. Kita akan siap-siap, pengumumannya akan sebentar lagi kita umumin, dan kita lagi revamp genome.dharma.or.id. Begitu nanti udah siap, kita akan segera umumkan di grup. Jadi di 2026 ini kita mau lebih fluid. Kita mau lebih banyak sequencing, kita mau aktivasi semua center genomic. Okay, I think that's all for me. Thank you very much and enjoy your day.
Thank you, Dr. Vincent. And we really hope that your vision comes true very, very soon. And for that, we all need your guys' collaboration. And the best way to collaborate is usually while talking to each other. So we have provided coffee and refreshments at the entrance area. However, since we are running a bit late, I would like everybody, if you would like to grab something to drink or to eat, how about you just grab it and then we bring it inside so we continue with our program, and then later on there's still going to be the networking dinner where we have enough time to talk to everybody and ask a lot of questions. If that's okay with everybody, if you need to grab a drink, please go ahead and then straight away return to this room so we can continue with the next speaker. Thank you very much.
Orang pada nggak kelihatan yang saya kelihatan itu.
Ladies and gentlemen, due to the interest of time, if you could kindly return to your seats. We would like to start with the next point very shortly.
Halo, habis baterai. Bapak Ibu sekalian, mohon bisa kita lanjutkan acaranya. Langsung ya,
Dear ladies and gentlemen, if you could please return to your seats. The next speaker is already ready.
I'm sorry, Professor. Yeah, ladies and gentlemen, please back to the seat because we want to continue for the next sessions of the single cell foundations model and precision medicines.
Anyway, I can
So-
start waiting there. They're joining. So I can start. No problem. Yeah.
So, dear ladies and gentlemen, it's going to be my great pleasure to invite Professor Jong Il Kim, MD, PhD from Seoul National University College of Medicine, a leading expert in genomic medicine and bioinformatics with more than over 200 scientific publications in human genome variation, cancer genomics, and next generation sequencing. That alone is an achievement. Wow. We will hear from Professor Jong Il about the single cell foundation model of precision medicine. Please welcome Professor Kim.
Thank you for your introduction. I think people are thirsty, so please enjoy your coffee or drink. I speak. Anyway. So it's my great honor to speak here in this meeting. So even Steve introduced briefly about single cell foundation model. I'll talk about what single cell foundation model is and why we need that and what we are doing currently to make a foundation model in Korea. Okay, let me start with the single cell revolution. In 2013, single cell sequencing was selected as method of the year by Nature Method. Since then, the field has grown incredibly fast. We can now look at gene expression in individual cells. This allows us to see cell diversity, rare cell populations, and disease-specific cell state that were previously invisible. Along with new technologies, data has been accumulating at an explosive rate. The Human Cell Atlas already contains over 70 million cells. If we include other platforms like Cell by Gene, hundreds of millions of cells have been profiled worldwide. These data have the potential to answer the fundamental question: what cell type exists in the human body, and what do they do? But this raises an important question: how can we use these data smartly? We already have a good example in using large-scale data smartly. smartly. So that is the foundation model. Most famously, the large language models like ChatGPT. The foundation model is pre-trained on a massive amount of data. It learns general patterns. Then it can be fine-tuned for specific tasks with very little additional data. This key idea is train once and apply everywhere. Why do we need foundation model in biology? The answer is simple. Biology has accumulated enormous scale of data, but current machine learning models are too simple. They lack the ability to make causal predictions. They cannot answer what would happen if questions in complex biological systems. Foundation models offer a way forward. Foundation models are now emerging in single-cell field too. Researchers have begun asking, can it do for cells what large language models did for text? The Human Cell Atlas Perspective published in Nature in 2024 proposed exactly this question, moving from the cell census to unified foundation model of human biology. Here is the key analogy. In language models, a word becomes token, or sentence becomes a sentence of token, a sequence of tokens, and the grammar becomes statistical relationships. In single-cell foundation model, gene becomes a token, a cell becomes a sequence of gene tokens, and gene regulation becomes network interaction. The transformer architecture works for both. Why is pre-training so powerful? When you have only small data set, the signal is weak and noisy. But a model pre-trained on millions of cells has already learned the language of the biology. When we apply it to a new small data set, it performs much better. It overcomes dropout noise and missing genes. This is the power of transfer learning. So how do foundation models actually work? There are 4 steps. First, we collect millions of single-cell sequence data. Second, we train the model using self-supervised learning, for example, predicting the masked genes. Third, fine-tune the model for specific tasks, like disease classification. Fourth, we deploy this model on a new data set. What you can do with these models? The applications are very broad. We can identify disease-associated cell states. We can predict how cells respond to drugs. We can annotate unknown gene function. We can stratify patterns to predict treatment responses. We can even model cell differentiation trajectories. But current models have important limitations. First, representation bias. As presented in the previous speakers, most trained data comes from healthy donors of European ancestry. Less than 15% comes from disease samples. Over 80% comes from European cohorts. Second, most of the models are the RNA sequence only. They miss epigenome or genome or protein data. So these models, like black boxes, their prediction are hard to validate biologically. So how do we build better models? Three things matter. First, scale. Bigger models train on more cells. Second, quality and especially diversity. We need disease-rich and ethnically diverse atlases. Third, multimodal interaction. RNA alone is not enough. We need epigenome, proteome, and spatial data together. These are the directions the field is moving forward. Okay. So we are, I'm a biologist. My major is genomics and bioinformatics. And we are doing collaboration with the computer science, the expert in computer science and AI experts to make foundation model, single-cell foundation models in Korea. my role in the project is providing the data to overcome the limitations I mentioned before. So diverse and better data for the better foundation models. So I'd like to introduce what I'm doing in our lab to contribute this project. The first project is SCADE, the single cell atlas of immune disease. This logo is made by Dr. Seung Lee. He was the previous grad student in my lab. Now he's in Yale. This symbolized the Korean traditional multicolored paintwork found in palace rooftops. This symbolized the cells and the interacting antibody of the immune system. This slide is from the 2022 HCA General Meeting. It shows the roadmap, the Human Cell Atlas. Currently, the data currently, the data is the atlas is based mainly on healthy donors with limited ethnic diversity. But the vision is clear to expand toward disease course and greater geographic and ethnic diversity. Our SCADE project fits perfectly into this roadmap. This is the photo is from the last year's HCA General Meeting in Singapore. The slide was presented by Sarah Techman, one of the leaders of the Human Cell Atlas project. You can see the SCADE, like over there, listed here as one of the major human disease atlases, alongside many different international projects. We are proud that SCADE is recognized as part of this global effort. SCADE is funded by Korean government, about $15.8 million over 6 years, from 2020 to 27. Over 120 researchers across 8 institutions are involved: immunologists, clinicians, and bioinformaticians. The project is integrated, organized around the 4 cores: clinical core, profiling and sequencing core, integration core, and mechanism research core. We selected 16 immune diseases across 8 different organs. The selection was based on these criteria, like left here, and including two pediatric diseases. For each disease, we profile samples from at least 36 donors up to 72 to capture individual variations. Importantly, we collect not only blood cells, but also disease tissues. This table shows our sampling strategy for each disease. We collect solid tissue, PBMC, and fluid samples depending on the disease. We use a wide range of profiling technologies: single-cell RNA sequencing, ATAC-seq, spatial transcriptomes, and more. All samples are collected across major hospitals in Seoul. They are very close to each other. In August 2024, we published a comment article in Nature. In this article, we described how regional projects like SCADE, relatively small in scale compared to HCA, can still contribute meaningfully to the global scientific community. We highlight 5 advantages of regional consortiums: geographical proximity for fast sample transfer, universal healthcare system, unified ethics and regulations, aligned national research priorities, and the ability to build local research network. We hope this model can inspire similar efforts, not just in Korea, but potentially SCADE Indonesia or SCADE Southeast Asia in the future. This is the roadmap of our project until 2027. And this is the example of the UMAP, the map of 2 million cells. This the version 1.0 open public in 2024. It includes data from more than 300 donors, over 500 samples. It covers 13 immune diseases. As I mentioned, current foundation models rely almost entirely on RNA data. But RNA alone cannot fully capture the complexity of the cell. We need multimodal data. Our second major effort is therefore to generate multimodal single-cell data at scale and at low cost. Looking at the trend Jacob cell by gene data set, you can see the transcriptome data makes up of 97% of all deposit data. Multiome data is only 2%. This is the critical gap. And most multimodal data comes from just a few organs, like brain, eyes, and heart. We need much broader coverage. In our lab, we have developed two in-house technologies to address this question. The first is single-cell multiome-seq, SCMM-seq. It profiles single-cell RNA and ATAC simultaneously with multiplexing of 8 to 24 samples at a time. The second is SC-Core-seq. It profiles ATAC and histone modification together, which was previously very difficult and expensive. We also used the Ultima Genomics platform to dramatically reduce the sequencing cost. I hope Jason and Steve will help us a lot. And specifically, I'm waiting the $10 genome period to make this model better. Here is one example. We applied our SCMM-seq to long airway organoids by integrating data from multiple technologies like single-cell RNA, single-cell flag, single-cell SCMM-seq, and long-read sequencing. We were able to identify cell types and gene regulatory mechanisms that were not previously known. This kind of multimodal integration opens new biological insights. We are now applying these multimodal approaches to scale samples as well. The goal is to build a truly comprehensive atlas with transcriptome, epigenome, proteome, and spatial data that can serve as high-quality training data for the next generation foundation model. So what comes next? The concept of virtual cell is emerging. Virtual cell is a keyword of the foundation model, single-cell foundation model of the next step. A virtual cell has three key functions. First, prediction. Given a perturbation like a drug treatment or gene editing, what will happen to the cell? We can make the data by experiment, or we can make the prediction model. And the next is discovery. What the new biology can we learn from the model? Third, explanation. Can the model tell us the molecular mechanism behind the prediction? Currently, the most experiment is done by manual experiment in each lab, but if we combine this strategy with robotics and laboratory automation, AI can design experiments, receive results, and design the new next experiment, creating a self-improving research cycle. This could revolutionize how we do biology. Another exciting direction is the digital twin. Current atlases build an average model of human cells, pooled across many donors. But in the future, we want models that are specific to each individual, incorporating their genome sequence, lifestyle, and environment. Imagine this. Instead of finding a drug that works for Alzheimer's disease in general, we can find the best drug for a specific patient with minimal kidney toxicity, considering his or her genetic susceptibility to kidney diseases. This is the promise of the digital twin. We are moving from population-level models to personalized and patient-level models. Okay, this is summary, but I heard that we are quite behind the schedule. I'll skip this slide. Okay, the work would not be possible without my wonderful team. I would like to thank all current lab members, alumni, and collaborators. Special thanks to the SCAD consortium partners across 8 institutions in Korea. Okay, thank you for your attention.
Okay, thank you, Professor Kim, for such a really good, insightful from the Korea. And now, next, our speaker to connect a population's genomic with one most important priority is in the public health, which is as a cardiovascular disease. The sessions remind us how the genomic is not only future possibilities and also a practical pathway toward early understanding about the risks, better preventions, and also more personalized care. Please welcome Dr. Peter Say, MD, PhD, as investigator of Emeritus in the National Health Research Institute and the chair professor at the Taipei Medical University. A pioneer of human genetic cancers and genomics, he has played a key role to building the Thailand's genomics research infrastructures and also leading the major precision medicine initiative across Asia. Within distinguished career, spanning over 3 decades of including fellowship in the MTSNA and the Harvard Medical Schools, his work has contributed important discovery in heritage disease and also the cancer's genomic. Today, we will present Dr. Say. Please welcome.
Thank you so much. First of all, I would like to thank Dr. Jason Kane and Dr. Stephen Lee to inviting me to this very important and meeting. This is my first trip to Jakarta to visit this great city, and I began to learn more about the diversity of this country. So I'm going to present my work on small scale, but I think I would like to give you some flavor on the medicine. So my topic is about the integrated genomic prediction. So I'm going to use a cardiovascular disease as an example to elaborate to you about the importance of population genomics and its relevance to precision medicine. So this is my disclosure. Oh, how do I do that? Okay, so I'm actually wearing 2 hats. So on one hand, before I retired last year, I worked in National Health Research Institute, NHI for short, for over 25 years, a quarter of a century. This institute was established 30 years ago. So we have a headquarters in the Hsinchu area. So this is the Silicon Valley of Taiwan. The very famous chip foundry TSMC has a lot of factories in that area. In Taipei, the capital of Taiwan, we have a business office there. And down in south, we also have a clinical facility in the city of Tainan. Taiwan has put a lot of efforts and investment on the genetic resources. This is one of the 2 papers published in Nature, October last year. In these papers, the authors from 16 medical centers throughout Taiwan, which include 33 affiliate hospitals working together to use one single SMP array, which allow them to get sample for 500,000 people and genotype of all these recruitment and the participant on the hospital base. I knew SMP array is very soon become obsolete, will be replaced by genomic sequencing, in particular whole genome sequence. But in very short period of time, this effort led by Professor Pui Ko were able to get a good sense about many polygenic risk for at least over 200 disease. Right. I'm the founder of this company, Product Development Center for Precision Medicine. So this company was co-founded by my colleagues, Professor Rob Kirby when he worked in Taiwan, and also an advisor, Dr. Mark Dundenberg from Chicago. So the company was established in 2022, April of 2022. So this company is now 4 years old. It is a spin-off company from NHI. So while I'm an investigator in this National Research Institute, we spin off our team and technology to establish this company. A main purpose of this company is try to build the so-called PPP, the public-private partnership. We want to utilize the public resources for infrastructure, but on the other hand, we would like to take advantage of the flexibility and the competitiveness and efficiency of a private company. The goal for PDC4PM is to uncover a lot of the genetic variant which is underrepresented for the Asian population. So this is a very important slide drawn by myself and also Professor Rob Kirby when he worked as a professor in my university. So imagine that we have many hospitals like in Korea or in Taiwan or Japan or Indonesia. There are many doctors working on lung disease or rheumatology. They all have their own samples, but each one, the sample size is small, and the clinical data differ from from hospital to hospital. It would take the efforts of a national efforts or a centralized the organization like NHI where I work, and also the Academia Sinica, which is a government sponsored research institute. The paper I told you, the Nature paper, was organized by Academia Sinica. So what the PDC4PM and other private companies serve is to form a consortium of the private sector, which through a formal agreement between national NHI and Academia Sinica, we can help manage and govern the data, and also to make the best use of this data for biotechnology or even pharmaceutical development. So this is the basic idea of the so-called Taiwan Whole Genome Sequencing Consortium, TWC. Right. So what does TWC do? We want to identify important disease area and to apply the very powerful whole genome sequencing technology to look into specific disease category. So we are taking a patient-centric, disease-oriented approach to work on important public health issues. So what are the important public health issues? So as pointed out by WHO, the so-called NCD, non-communicable disease or chronic disease, is the most important cause of death and also the most important burden for socio-economic wise. So we just exclude those epidemic like pandemic like SARS-CoV-2. 75% of this planet people died of non-NCD. So the NCD or chronic disease, The most important one is cardiovascular disease. Also, we can consider that a lot of cancer, breast cancer, prostate cancer, colorectal cancer can become a chronic disease. Also, diabetes and lung disease are the important category. So within our company, and also working together with NHI and quite a few medical centers in Taiwan, we have established a practical way, which is not as powerful as whole genome sequencing, I have to admit. But at least until now, we have proven that this integrated genomic technology can predict the genomic risk for certain disease. So I would like to add at least one technology, which is proteomics, onto this genomic, and which we include whole exome sequencing and the low pass or low coverage whole genome sequence. Let's review again. So SNP array is relatively cheap, inexpensive, but it allowed the Taiwan Academia Sinica to recruit 500,000 participants in very short period of time and complete the genotyping, although the information is very sparse. In other words, this array only scan or interrogate 0.7 million bases of 700,000 single nucleotide polymorphism. On the other hand, by contrast, the whole genome sequence is 3 billion bases. Exonic sequence is like 1% or 2% of the genomic content. So we came up with this idea, but we are not the only one. The Broad Institute, the clinical division of Broad Institute, has also come up with a similar idea. They call it a BGE. BGE stands for Branded Genome Exome. So by combining the power of deep sequencing for this exonic sequence and also the low coverage of the whole genome, it would allow us to identify most, if not all, of the genetic content that are relevant to the disease processes. One key point, though, is I kept thinking that because at the beginning, I truly believed that whole genome sequence will be the eventual goal. Okay, it was not affordable maybe 3 years, 5 years ago until the newcomer, for example, the Ultima Genomics technology come along. We evaluate that the data size for whole genome sequence would occupy 80 gigabytes. On the other hand, if we use whole exome sequence plus low coverage, it will only 12 gigabytes. So it will be easier for a small clinic or small medical center to handle the IT issue. Imagine if you have 100,000 cases, each one has 80 gigabytes. It will become a big issue for a small research unit or clinical facility to manage. So in summary, the whole exome plus, which combine the whole exome and low coverage, can identify most of the pathogenic variant. It can also work on identify some of the structure variant. It can offer information about polygenic risk score, so it's cost effective and efficient. So I would use the next 5 minutes because I know I'm standing between you and the dinner. So I would take this important disease, hypertrophic cardiomyopathy, HCM, as an example to show you how we have been using this low coverage whole genome plus whole exome to understand better about the HCM in Asian, in the Taiwanese patient population. So we are about to put up a manuscript. So this is what we found. So we found a distinct genetic landscape of this disease. It's quite different from what have observed in Caucasian, in white people. So the Asian people have different, almost totally different genetic landscape in this disease, very important disease. So that illustrates the importance of population genomics in the context of precision medicine. This is a picture taken from a review by Professor Eugene Brownwell. He's the father of genetics of the cardiology. Okay, so on the left-hand side is the normal anatomical structure of the heart. You can see clearly about the heart muscle which pump the blood, right? And we can see the left ventricle and right ventricle, and the regular structure of the sarcomere, the basic muscle fiber. On the right-hand side, which is an anatomical picture of a hypertrophic hypertrophied heart, you can see the thickness of this interventricular septum here. It's become really thick, okay? And the chamber of the left ventricle become relatively small. So in other words, this pump become very inefficient. The only small amount of blood can be pumped out. Therefore, the patient will have heart failure, dyspnea, and all the symptoms associated with poor oxygenation of the body. Okay, historically, under microscopy, you can see that the underlying cellular mechanism is the arrangement of the sarcomere structure. Clinically, HCM is the most important genetic disease of the heart. It can lead to 4 different outcomes. About half of them will be fine throughout life, but a very high proportion of them will end up with heart failure. And even more serious is some of them will have developed atrial fibrillation, stroke, or even sudden cardiac death. So the clinical picture will be different, even you have the same disease. So this slide summarizes the important finding about genetics. So in the Western population, in Europe, in the United States, about 40% of the cases with hypertrophic cardiomyopathy, HCM, has a non-single gene mutation of this A sarcomere gene. So by screening this A gene, one should be able to identify quite a few of patients who has this underlying single gene, and from there, we can also do the so-called cascade screening to screen for the relatives who might also carry this disease, even though they don't know that they have the disease or they don't even develop any clinical symptom. All right. I have added this slide after I came back from the symposium from Korea. So I cite this very important comment by Eugene Brownwell, Professor Brownwell. He's more than 95 years old, okay? He's still quite active. So he quote, "The routine application of genetic testing and preclinical identification of family member represent an important advance." That's a very important advance for genetic in medicine. Moreover, the genetic discovery have enhanced understanding of the molecular pathogenesis of HCM and has stimulated efforts designed to identify new therapeutic agents. So not only we could now understand clearly the molecular basis, the genetic cause of the disease, we can also begin to design precision medicine, the right drug for the right patient. In our group, we have collaborated with one of the best heart center in Taiwan. So far, we have conducted over 200, we have collected, recruited 261 patients from this center. So most of the patients, they have short of breath, so they will have an echocardiogram. So we can take a very objective measure by measuring the thickness of the interventricular septum. For those who are not cardiologists like myself, so it is important to remember that when the interventricular septum, IVS, is more than 10 millimeter, that's about the normal range. So we have divided the patient into 3 groups. One has undergone a surgical operation, which had include 26 patients. Those are the most severe cases that require operation to cut off some of the heart muscle to enlarge the chamber. And in the middle are the cases with IVS, interventricular septum, over 15 millimeter. This is the classic definition defined by American Heart Association as a clinical definition of HCM. Some of them are at risk, which is over 10 millimeter. So those are represent most of the cases, even though they are not severe enough, but they have clinical symptoms. All right. So without going into the detail, so we have completed the analysis by both the whole exome sequence. We examine only those gene 70 something related to the heart gene, and also we include some of the gene that related to familial hypercholesterolemia to do the whole exome sequence analysis. Also, because we have conduct low coverage whole genome sequence, we were able to get the polygenic risk score for this patient. So in this poster, which was presented by my student, Kenny Chen, I would like to that remember in the white people, Caucasian, HCM has 30% of the gene that is monogenic, familial. But paper published in Singapore, they report only 16.5. That's 16 and lower. But in our population or in our cohort, only 6.5%. On the other hand, we could find some variant of unknown significance, VUS, which is uniquely present in Asian people, some of them in the Southeast Asia, some of them also cover Japan and also Korea. So there's small difference between the population of Asian people, but these 2 variants are only present in Asian. So that's why I think the population genomic is very important. For otherwise, if you get this sequence, if you look at the CLIMBA, which only document the data from white people, they will say, oh, this is a benign variant. By contrast, we identify 8.13% from our patient who actually has a different disease called Fabry's disease. So in southern parts of China, include Vietnam, Taiwan, or even Thailand, there's one intervening sequence variant that occur very high frequency in Taiwan, 1 in 400 female, 1 in 800 male, because this is a X-linked trait. Having this variant of IVS4919 will cause the reduction of alpha-galactosidase, which is the causes of Fabry's disease. So these patients, even there are many of these cases in Taiwan, but not all of them will develop a heart problem, but they have definitely increased risk. But by identifying those cases, which is represent 8.13% of the cases, they can be treated by enzyme replacement therapy. That is totally different from the classic SCM. Because of time, in the interest of time, I will not go through with you about the importance of the additional information we could find with PIS, because although we can only find 16 or 6.5% that is monogenic, but we can use the polygenic risk score to identify those cases that has high risk of developing hypertrophic cardiomyopathy. Also on the clinical side, the higher the risk score, the more severe the clinical manifestation. So we were very excited that the PIS not only predict risk, it's also correlate with clinical severity. All right. So why I become so fascinated with the power and the technology of Ultima Genomics. So I think before you listen to Dr. Stephen Lee's talk, I knew about that Jason is going to join this company. So I take a look, because I have been a long-term user of the other competitors. So, but in order to generate big data, I think it's very important to have some technology that is reliable, efficient, and affordable, right? So here is my proposal to Stephen and also Twist, that perhaps together, by taking the advantage of generating libraries in a very efficient way and affordable way, and also the power of sequencing, we can together generate a lot of big data for all the clinical relevant project we are going to pursue, right? Because we realize sequencing per se, the machine and the reagent, the cost has become smaller and smaller. Although we have heard very encouraging news that the material cost will drop significantly from 100 to 80, 60, maybe $10 per whole genome. But there has been a lot of discussion where is the bottom? Okay, trust me, $10 will be the real bottom, because still we need to make the library, we do need to analyze the data, we need to store the information, but the major cost of the sequencing per se, which is used to be very, very high, which will become a major factor, is no longer a big problem, correct? So we have at least 2 projects in process. I have talked to the folks of Ultima Genomics, One project which we have been conducting is on the aging process. Okay, Taiwan, Japan, Korea has a very serious problem of we are moving into the super old country, which by definition that 20% of the population of Taiwan is 65 and above. And myself already contribute to that 20% because I already turned 70, believe it or not, okay? But in order to understand what's happening with regard to chronic disease. So the heart disease, the diabetes, the kidney disease become more prominent with aging. Okay, so at NHRI, we have established a cohort for aging study called Healthy Aging Longitudinal Study of Taiwan, HALST. It was established in 2009, 17 years ago. And these individuals, over 5,000 of them, have been followed up regularly with longitudinal data. So it was great, have all the biochemical clinical data. But I approached the group leader, of course, and tell them that what's the shame if you don't have genomic data, then you cannot make the best out of this cohort. So with the strong support from the company, the government, we were now able to complete 50% of this cohort. So we now can bring together the genomic data with those clinical observation, the clinical data for diabetes and chronic kidney disease. So my dream is to expand this cohort to lower the age to not 55, to 25, 35, 45. We can get a lot of genomic data to do this type of study. All right. The other topic is on liver cancer. Hepatocellular carcinoma, HCC, is a major cause of cancer deaths in Asia and Africa. We were delighted to know that between the Twist and also the Adiwan Genomic, they have a lot of new tool, new reagent, a new solution for doing liquid biopsy. So since we have collected a big cohort on biorepository of liver cancer in Taiwan, so we are moving into this direction. So we're going to take the surgical sample, we'll sequence the germline and the tumor. Also, we're going to collect the plasma sample before surgery, 3 months, 6 months, or 3 years after surgery to monitor the early sign, the early sign of recurrence, because HCC, liver cancer, is notorious for its recurrence. 1/3 of the patient will come back with a new tumor. Okay, so monitoring the recurrence and also to design specific treatment when the cancer come back is very important. Also, that should be included into the clinical practice. Before I came to Indonesia, I asked myself, what do you know about the public health issue of this country, which has 218 million people, and what's happening in the world? Okay, it turned out that hypertension surprised me. That is a global health issue. Canada, Taiwan, and Indonesia all face this chronic disease with aging population. but each country has different challenge. For Taiwan, for Korea, because the hypertension become important because blood pressure tend to become higher with age. Okay, but on the other hand, Indonesia has even higher percentage. I read about 40% or so of the population, and a lot of them are young patients. And considering the population size, I think that will be an important issue that we can work together with Canada, with Taiwan, Korea, Japan, or Indonesia. So we have discussed this possibility with Jason and Stephen. So we propose to do IGP, integrated genomic prediction. We'd like to at least collect 6,000 cases. We would move from whole exome plus to whole genome sequence. Now the cost is no longer a major issue. I think the key is to identify carefully those patients, certify them, and monitor and collect information about the treatment, the outcome, and so on, so that all these can be put into part of this foundation model that we can better manage this disease of global importance. So I would like to conclude that even we have made good progress to design this IGP, integrated genomic prediction, using whole exome and low coverage. But I think it's time to move on to the new technology or new possibility of optimal genomic, which we could routinely offer whole genome sequence for all the disease category. To me and to my company and my institute, we focus on disease cohort, not the community-based biobank. Okay. I would like to end my talk by acknowledging the very capable student, Kenny Chen. Okay, so he's about to get his PhD, and he has 3 publications ready to be submitted, or 1 already submitted. And also Rachel Lin, who did the host cohort, and a very capable technician who look after this liver cancer project. And also, of course, the support from Ultima Genomics and Manning Medical Center and the patient. So I would like to extend my sincere hope that I really want to work with scientists in the Asia Pacific region, Indonesia, Korea, Japan, and Thailand, and so on. So together we can fill up this gap, which is lacking in the global situation of population genomic and precision medicine. If you have a question or you have any interest, please reach out to me. Thank you so much.
Thank you very much, Dr. Chai. And yeah, I very much do hope so that we can collaborate together in the future, and that also tonight you will find a lot of other people to collaborate with. Yeah, so now we come to our last speaker of this scientific event. Therefore, I would like to invite Dr. Jim Zhang, PhD, Senior Field Applicant Scientist at Twist Bioscience. He holds a PhD in genetic from the National University of Singapore and brings extensive experience in next generation sequencing across research and industry. Today, he will present Writing the Future with Twist Bioscience NGS Solutions. Welcome, Dr. Zhang. I think they're looking for the presentation.
Actually, this is perfect timing. We're gonna have some butter and other like drinks come up to the table while we're waiting. So please sort out the presentation, uploading. Hotel staff, please set the table so we can have dinner right after this. Oh, actually, we're gonna take group photo from here. You don't have to get up. And then we're gonna have dinner, right? Yes.
Oke. Jadi kalau staffnya mau anterin
Silakan.
minuman dan lain-lain, silakan.
Udah boleh mulai setup button.
You already found the presentation? Yes? Okay, then let's work. Apologies for the technical difficulties. Or maybe while we wait, can you tell us a bit more about TWIST for the people which don't know anything about what you do? Please.
Yes, definitely. So my name is Jim. I'm an application scientist at Twist Bioscience, right? So you may be wondering, why is Twist doing at an event that is hosted by Ultima Genomics, right? So in the talk that I will give later, I hope to kind of give you an understanding and impression, how Twist and Ultima are two really similar companies, despite, the difference that they may seem to be, like two different technologies focusing on different solutions. As Professor Peter has mentioned, right? There are certain similarities and, synergies that we can unlock by combining technologies from Twist with Ultima, right? I would say Twist, we are operating somewhat in the upstream. Yep, here's the slides. And we're getting really close to dinner, right? Beef tenderloin and chocolate cake. But I think we started off this seminar with a great introduction from Dr. Stephen, right? Ultima Genomics is a technology company that is really built from the ground up. We're not interested in incremental improvements, 1%, 2% improvement in performance, reduction in costs. We're interested in 50%, 80%. And in that way, I think Twist is really aligned with Ultima in that regards, right? We're not here to do incremental improvements. We're here to disrupt technologies. We are here to provide solutions to problems, genomics problems, population level genomics problems that cannot be solved with the technologies that we have today, right? Or we used to have, right? And like Ultima Genomics, Twist Bioscience is also a US-based company. We are located right here in, that's not really working, but yeah, we are located in San Francisco, right? We are 13 years old now, founded in 2013, and that's where our headquarters and also where we produce all our NGS reagents and oligos for target enrichment. It's just a 5-minute drive away from the city. That is an important point. And somewhere to the north, 2 hours by flights, we have another facility, right, in Portland, Oregon. That's where we produce synthetic biology products, like custom genes and gene fragments, for example, another part of our portfolio. But really, I think what this slide is showing is really emphasizing the global presence of Twist Bioscience in different regulatory markets, right? In places where NGS platforms, they prioritize different platforms, right? But we exist in all these markets. I think that really speaks to the quality of the products, the disruptive nature of the technology, And this is also very important because it does give us all the valuable feedback from different regions of the world and allow us to become a global genomics provider. And now I talk about, we're here to disrupt the market. This is how we are doing to disrupt the DNA synthesis market, right? So DNA synthesis has been around for quite some time now, maybe 50, 60 years. And how Twist disrupt the market? Like Ultima, we also rely on semiconductor-based silicon technology. Our CEO is not a biologist either. She has a chemistry background. And this is fundamentally ground changing, right, in DNA synthesis space, because when we synthesize oligos on a silicon chip, and these tiny little droplets, they are called nanowells, and each droplet is 1,000 times smaller than standard DNA synthesis reactions, right? When you have smaller reactions, things really get easier because it's easier to control the temperature. Silicon is a great conductor of heat. And also you can control synthesis reagents very precisely as compared to conventional way of DNA synthesis, right? You are using 96 well plates or 38 well plates. Reaction volumes are much larger. And that is the kind of thing you can get from a very traditional synthesis because each reaction is independent of one another, right? Whereas on a silicon chip, up to 1 million oligos are synthesized in parallel under the same exact conditions, right? So what you get is a very uniform pool of oligos. And what this means, at least for NGS, is that when you use them for example, target enrichment sequencing, the quality of the reagents has a direct impact on the quality of your sequencing data. And that's what this slide is trying to show, right? The fact that if you are trying to sequence an exome, for example, you're going to get, that uniformity that you need, right? All targets being uniformly captured. You reduce under sequencing, you reduce over sequencing, which are all wasted sequencing at the end of the day, right? And what do we do with all the oligos that we make on our unique silicon-based synthesis platform, right? Firstly, they get formulated and they get shipped in oligo pools, as its name suggests, pools of oligos, right? Which we can do a lot of interesting research. For example, pools of oligos can be assembled into gene fragments, can be cloned into vectors, so we get clonal genes. And that, in fact, has been a very important tool in the last few years, right? When we talk about mRNA vaccine development, antibody genes, all of those have to be expressed in some ways, right? Using DNAs, right? That DNA clonal genes that Twist manufactures, right? And then we move on to site-saturated libraries, combinatorial libraries. So these are just different ways of engineering proteins, right? We can put in, we can like tweak a single amino acid. We can change at one site. We can also do them on multiple sites. What this presents to us is a combinatorial challenge, because the more mutations you try to introduce into proteins, the more complex your libraries are going to be, which means you need like millions or billions of oligo custom vectors. Where are you going to get all that, right? It all goes back to how we synthesize the oligos. Now, why am I going into all this, right? It's because of the fact that the ability to synthesize DNA at a very high throughput allows Twist to evolve to become a protein engineering company, right? And the proteins that we engineer all end up in our NGS portfolio, which, right? Just a very quick run through of the overall NGS portfolio that Twist supports, right? We have synthetic controls, we have library prep, we have target enrichment, and also fixed custom panels. All of these, right, fit one way or another into sequencing platforms, right? And of course, as we hear from Professor Peter, right,
some of these very new chemistries are being validated on Ultima Genomics platform right now. We already have validated solutions, PPM-seq, as Dr. Stephen introduced, are fully validated. Twist, Ultima collaboration, disruptive technologies coming together, very powerful tools for us to use, right? So I'm not going into the details, except to say that, you know, we talk a lot about upstream, library preparation, all the work that you have to do before you can sequence on a UG200, for example. Library prep at the end of the day is all about enzymes. If you think about it, you need to fragment the DNA, you need to ligate adapters, you need to do PCR. All these are driven by enzymes, and the quality of the enzymes has a direct effect on the performance of the workflow. And Twist, being able to synthesize the DNA, being able to express the proteins, being able to engineer the proteins, this is what we call a vertically integrated structure. When you control the entire manufacturing chain, there is a lot of magic that can happen, right? Because you are no longer a small player in the workflow, right? Where you have limited control, right? When you control the whole workflow, right? You can introduce disruptive change. And that really ties to the next point, right? Application driven, because, you know, genomics research is not straightforward, because we heard about NIPT, we heard about cell-free DNA, liquid biopsies, and we have FFPE tissue samples stored in the archives for 20, 30 years. All those, you know, introduce different demands for NGS workflows. And that really ties back to the enzyme engineering capabilities from Twist, right? Because we engineer a unique solution for the specific problem that we are trying to solve, right? Custom solutions. So I want to take a moment. You may have seen the Twist poster, True M Library Prep poster. So this is the outcome of several years of R&D, protein engineering work by Twist Bioscience, right? So True M is, you know, originally in its previous form, it was more like a bread and butter kind of library preparation solution, right? Good for most sample types, and, you know, it works robust. It's not too expensive. But we were thinking, you know, how do we engineer this bread and butter library prep kit to have best-in-class performance? And that's where we really drill into every single step where enzymes are at play, right? I'm supposed to have an animation here. Oh, yes. So starting from fragmentation, right? We have DNA fragmentase that goes in and cuts up your DNA, and we want that fragmentation step to be consistent, to be tunable, so that you consistently get the fragment size you want, because different sample qualities can end up, you know, giving very different fragment sizes, and you have to have 10 workflows for different sample types, and that's really hard to manage. Next thing is ligation efficiency, right? By ligation efficiency, we're talking about how efficient can we attach the sequencing adapter onto the DNA molecules. Each DNA molecule that fails to get an adapter fails to get sequence. So whatever sequence information is on that molecule is lost, right? If it's the cancer-causing mutation, that is lost. If there's a, you know, basically we want to maximize that step, and the way we do it is to make use of a engineered ligase, right, in this kit. And last but not least is PCR amplification. It's a new polymerase also engineered by Twist. So what I like to think about this is, what we're trying to do is to take a very bread and butter library prep kit. Think of it like a Toyota car, very mass market, very robust, very cheap, and engineer it to give us Ferrari-like performance. And we compare that with a very prominent competitor product out there in the market. I shall just call it competitor K, very well known, maybe Kit K, owned by competitor R. But anyways, this is a very well known product. Maybe we can consider it as a Lamborghini. And look
At the performance in green, our modified Toyota beats the Lamborghini on all metrics that we measure, right? Whether it's a concentration, library size, complexity, and mean target coverage, all this coming from the engineered enzymes that we use to maximize ligation efficiency, maximize PCR amplification efficiency, all that translates into higher mean target coverage at the end of the day, lower % of zero coverage targets. What this means for us as end users is that at the end of the day, right, you can process more samples, right? Because samples that would originally fail the existing workflow from product K now becomes highly usable, right? You can process it downstream, you can sequence it, you get usable information, you get better data. And when, combined with very promising technology from Ultima Genomics, you lower your overall costs, right? So you get the best of both worlds, right? Upstream enzyme engineering, downstream wafer-based sequencing, and what comes together is, I'm sure, what is motivating Professor Tsai in, driving a lot of his population-based, disease-based screening projects, 6,000 samples. All these things were, you can do them previously, but it would be extremely cost prohibitive, right? Fragmentation. For any of us here who works with NGS library preparation, controlling fragment size is always a pain because samples come in all sorts of qualities from different extraction kits, from different sample sources. We make it such that with the engineered fragmentase, it's very easy to kind of tune the fragment size. In fact, our R&D team has managed to kind of measure it to the extent that, 1 minute of the fragmentation time causes a difference in 50 base pairs of the final insert size you get. That's the level of precision and accuracy that we can get and the tunability that we can get with true M DNA library prep. So once we have set on a specific fragmentation time, right? Sorry about that. You can be assured that that condition is going to work equally well regardless of the DNA input, regardless of the DNA quality. So we have validated all these, right? Once you have identified a 22-minute incubation time and you have 6,000 samples, all of them are at different qualities to you. Does that mean we have to optimize 6,000 times? That's not the case with TrueM. You can get very nice, even, consistent fragmentation. One condition, whole range of different sample qualities, input, and even the sample types, right? So there's some pretty disruptive advantages there. And the good news is that, this kit right now is, we're developing full-length Ultima UG barcodes for this library prep kit. It's a PCR-free library prep kit that utilizes the fragmentation module as well as the ligation module. We don't need the PCR polymerase because, we don't need it, right? PCR can just introduce a little bit more bias. So this will be available probably later this year for the latest UG 200 platform. So definitely something to look out for. And so when we look at library preparation, another problem that we have observed, especially for population scale genomics, is that, if you have 96 samples and you want to sequence them, it does mean that you have to make 96 libraries. You have to quantify each of them. You have to dilute them. You have to fragment, ligation, clean up, PCR, clean up again. You have to do all these 96 times. If you have to do 1,000 samples, you have to do it 1,000 times. So you can imagine this is really not a very scalable method of doing NGS, right? And your costs increase directly, linearly. So we looked at this workflow and we think, how can we make things more cost effective? How can we make things faster? The first step, quantification and dilution. You have to use, go to the lab, do the measurements, plot all the figures in Excel files and then go back to the lab and one by one tune your pipette. It's not fun, right? It's not fun. I had my fair share of doing quantification and dilution. There was no flex prep when I was doing my postgrad, but the technology is available now. It's called normalization by ligation. We use a modified adapter, which we synthesize, synthetic DNA, to do the normalization for us. What this means is that your DNA input can be of different levels of input, 30 nanograms, 300 nanograms. We don't care, right? It just goes into the workflow. The enzymes, the adapters do the normalization for us. So immediately after ligation, what you get? Normalized library. The second thing, instead of having to do 96 times of all samples across the whole workflow, can we somehow condense the workflow somewhere in the middle and leverage on, pooling efficiencies? And that's what happened, right? With FlexPrep, the adapters also contain barcodes, right? So this is what we call inline barcodes. So they enable samples to be pooled immediately after ligation. So as you can see, right, if you have 96 samples, relatively early in the workflow after ligation, you are left with 8. That immediately means 12 times less reagents, 12 times less effort, 12 times less even consumables like PCR tubes and pipette tips. All those add up at the end of the day, right, to really drive down that disruptive cost savings that we were aiming for, right? So we're not going to cut it with just, improving ligation efficiency by 5%, none of those things are going to help, right? So, and also the fact that because the only instrument you pretty much require is a PCR thermocycler, it's extremely easy to automate as well, right? So here's just the results that we can expect. Once again, whether your input is 30 or 300 nanograms, we're talking about 10-fold difference here, right? We don't care about that. We just put it through the workflow. The technology do the work for us. And what you get is, it's not perfect, right? It's not perfect. But considering the scale of the input that we are putting in, I would say we get very usable data. This is not just convenience, right? It's not just convenience of bypassing quantification and the normalization. This does means that even with samples that are vastly different, right, you end up with the same kind of representation, data output that allows you to directly compare between all samples, right? So you do not compromise on the data quality, right? On top of the cost savings that the workflow provides. And this is actually my last slide, which really adds on to the point that Dr. Stephen mentioned, right, with regards to extremely low mutation detection in a very challenging sample called cell-free DNA. So we have implications in cancer detection, NIPT, for example. I myself am really impressed by this technology from Ultima, right? Disruptive technology, where we actually physically link the top and bottom strand of the cell-free DNA molecule together. We preserve that strandedness so that the concordance of the mutation can tell us, is it a true variant or is it an error? So very elegant, very easy way to correct PCR and sequencing errors. It does require the adapters to be attached to our cell-free DNA at very high efficiencies. And once again, that's how the engineered ligase comes into play. Once again, we benchmark against 2 top cell-free DNA solutions in the market, and still we are getting about close to 10% increase, right, in the percent of duplex reads, usable reads that can allow us to correct the PCR or sequencing errors. That really translates to around 10% increase in mean coverage, which means 10% savings in cost. It all adds up at the end of the day, and also better data quality with reduced NGS library prep artifacts as well, right? So end of the day, maximize data output, minimizes artifacts, cost effective, sensitive, and think this is my last slide. Yes, thank you.
Thank you very much. And if you don't mind, please stay on stage, because I would like now to call all the other remaining speakers also back on stage. Dr. Stephen, Professor Kim, Dr. Peter, yes.
Yeah.
And
I also kindly invite Dr. Vincent and Mr. Jasons to come up to the stage to present for the souvenir. Dr. Vincent first. To Jason. Okay. Yeah. Jason, please.
please, yes.
Of course. You also get a souvenir, Jason. Yes.
Just give us some time to take some pictures. Okay.
Thank you.
Now, yeah, to Stephen.
Okay.
To Professor Kim. Yeah. Yes, please. Yeah, Dr. Jimmy. And now just invite to the all speakers into the stage, come up to the stage and take a pictures together.
Thank you very much.
Thank you very much for the speaker and the supporting for this event. And ladies and gentlemen, before we conclude of this event today, we would like to invite everyone on the stage to the group photo sessions. Yeah, I mean like we want to see to the screen and please the photographer to come up to the stage and prepare for the photo group.
Please stay seated. The photographer is gonna just switching his camera.
I will have to count 1, 2, 3.
So,
Sampai nggak? Kalau nggak sampai bangku, bisa pakai bangku. Oke. Naikin satu bang. Coba kalau kamu dari sini pakai bangku, dari tadi.
Oke. Sorry.
so 1, 2, 3, smile.
I think from this left and right, they cannot capture from the. I think we need to a little bit. Oh, let me try to. Kayaknya setup terus ini aja nggak bagus. Tampilnya di sini. Karena yang di sini nggak dapat, jadi dilurusin aja sebentar. Itu di belakang. Ke belakang ya. Karena nggak dapet dokter gimana? Miring aja. Ya udah. Coba Pak diaturin dulu. Miring. Ya. Dari sini. Dari sini naik.
Okay, we're gonna change the different angle and try to get it from here.
Because we try to get a better and all the attendees can get the captures to the pictures. Okay, from the media can just join to the back of this table, so it can also capture the pictures. Okay, thank you, ladies and gentlemen.
Okay, the future of genomics will not build through presentations alone. So we still have to build collaborations. So therefore, we would like to invite everybody to join us for the networking dinner now, which will be served shortly.
Yeah, and close today's symposium, may we leave it as share sessions to propose the pathway to forward genomic inclusivity, precision medicine, and our requirement visions, trust, and collaborations. And today has shown that we will build a future already present in this room. Before we conclude, I also want to extend our sincere appreciations to organizations committee, including Pepsi, from the U.S. Embassy, Army, and entire of the YSDS team for their dedications for the schemes.
Our gratitude also goes out to Pak Urusis and the team from the Grand Hyatt Jakarta for the excellent support and the making of this event possible.
Thank you for being the part of important moments.
So please enjoy your dinner and continue the conversation. Terima kasih.
Terima kasih. See you on the next symposium.
Can I just recognize Master of Ceremonies, Ms. Novi and Dr. Nani? Can we have a great round of applause?
Thank you.
Thank you so much. You guys done a great work. Great job.
Okay, ladies and gentlemen, I would like to remember you that your souvenir and goodie bags are available to collect in the registrations deck. So remember, if before you leave in this place, you can take your goodie bag in the registrations deck. Thank you.